Hello. So the little talk we're going to do today on basically the way of some of the next. Oh, you look so sad.
No, I'm looking at Maria.
The way that the brain modifies its cortical structures in relationship to experience, which is kind of it's thought to be sort of one of the mechanisms of learning, and it happens in multiple different time points during development. That's probably what I'll talk about. Talk's not real long. It's chapter 24 from the Neurophsy text. And there's some very sort of crucial experiments that they went into some detail in. If you've taken neuroscience at an undergraduate or graduate level, you've gone through some of these experiments already. Okay, so how do we develop complex functions in our brains so that we can perform complex functions? There's been a bit of investigation into exactly how this might be going on, and some of the approaches we'll go through in a moment. But what we're asking is, how do we adapt the brain to perform its functions, which are often quite complex, like speech or visual interpretation, things like that. Some steps seem to be what are called innate, in that they are programmed even before any exposure and they exist without any real learning. So examples of this are certain sounds that animals can make, responses to, say, stress, like a fighting response, fight or flight. Those types of things are innate and you don't really require any learning. Some require some input, and the earliest examples of this are what's called imprinting. I'll show you a couple of examples of that in this one. And then some things sort of have both components, and this kind of the schema looks at basically what was easy to tease apart and doesn't really deeply address more complex things, such as say learning mathematics or music in a general way, and things that are very, very complicated are much harder to tease apart. So some learning do have both components that are, some are already wired and some are added on. And what has been found with experiments is that basically if you alter the input, and to some extent, if you alter sort of the consequences of the input, if you change some of the parameters of the system, you drastically affect how things actually develop during the development of the organism through life. Okay, so this is imprinting. I know what this picture is, so it may not be fair. This is a scientist, Dr. Lorenz, and behind him he has 3 geese lined up. And what he did was present himself as sort of the single object on which they locked onto during the first, I think it's 4 hours of life, but it could be the first day. So that they basically treated him as if he is their mother, and then they start following him around everywhere. This persists even if they're separated from him a while. They will start to lock onto other things that are, say, similar in structure, so maybe male figures, and they'll start following those things around. He comes back, they'll go back to him. There is a very defined period where this can happen for the species, for the geese. It's just that first day. If you miss it, it doesn't happen, and they don't imprint onto anything. So it's an example of sort of an acquired behavior that is triggered at a particular time and by a sort of a situation or a learning behavior. So the critical period comes into play with a lot of these particular behaviors that have been dissected and analyzed. And probably ones without great critical periods are just much harder to really tease apart, and that's why these are easier. Critical periods are basically intervals during which the imprinting or the learning can happen with the appropriate input. It's very time constrained for the following behaviors. That's on the order of 24 hours. And timing depends on the particular task, the animal, and also to some extent on what you do with the sort of the internal environment, which chemicals are available and which are not. You can manipulate that way also. Learning is possible at other times. It's not usually as complete, and we'll have some examples of that as well. Because these are some of the critical periods for different behaviors in different animals with particular sort of defined critical periods. So neuromuscular junction for the mouse, they looked at the learning behaviors for there, and basically a lot of the input and pathways are set up prior to day 12. As an example, for the taste and olfaction of a mouse, there's actually no critical period for this, but if the rat is any indication, then there's certainly some critical exposures that are involved. For instance, a rat will remember certain odorants from mom and will be selected with those later on in life with mating behavior. There's other examples here. The human example is absolute pitch. Apparently, somebody has somehow determined that happens before 7 years. I know one person who states that they have perfect pitch. I have no idea what happens to that particular individual before age 7 years, but somebody's done a study to try to sort of prove that it's locked in. It's not to say that all music happens before 7 years, but that particular component of being able to recognize a note absolutely without any reference is apparently locked in at some point. You don't believe that?
No, I'm saying I don't have perfect pitch, but I know some people who do.
Some people say they do, yeah. But it's usually violinists because you need
I
the
know a
pitch.
cellist and a violinist.
Could need it before 7 years.
It's developed.
You could probably develop it before 7 years, but there's a point where it becomes locked in. So after a certain point, you probably could. We'll go into the visual stuff in just a
You
second.
can work on it. You can like practice, like work up to it, so like you're pretty close, but it's not the same as.
Yeah, maybe sort of similar to that. This is some studies with infants that are either hearing or deaf, and whether or not they'll pick up on any what's called sign language babbling from their parents. I believe the parents for both sets of infants are actually deaf, and it's a question of how much they pick it up. I don't know, the deaf parents, how much sound they make, so that has some potential impact. Let's see where we're at. So with the deaf infants, since they have no other way of sort of interacting in a symbolic way with their environment, they pick up a lot of the mannerisms very, very quickly with a particular aptitude. You can see that the time they spend actually doing these activities is proportionally much, much higher. Whereas hearing infants who have other ways of potentially interacting, such as speaking, crying, and hearing their own speech, it goes down dramatically. So that was a human experiment looking at the behavior of the beginnings of sign language or sign babbling. This was actual looking at the different areas of the brain that light up in kids versus adults as they sort of develop language function. And there's sort of areas that are activated in kids, and they tend to be more anterior, whereas in adults, when they're performing language tasks, it tends to be a little bit more posterior. Why there's nothing in the middle, which is the actual functional language center, is a little curious to me, but I guess kids are setting up the connections, and the adults, these are more the established connections, is the way I'm thinking of this happening. And language seems to have a critical period also around 7 or 8 years, and there has been some experiments with individuals who are what's called feral. There was a woman who was raised by parents who basically restricted all auditory or language input. And so this woman did not develop language as a child. And then when they tried to teach her language later on, she barely got very, very limited use of language and not a lot of fluency. This was one sort of experiment that was done by, I guess, neuropsychologists.
But it wasn't zero language versus you have learned one language already.
No, that was not the case, but that critical period for language learning does apply to multilingual things. So your time to get your second language is before age 7 also. So if you don't get it before age 7, it becomes much harder. So there's some good to all the pushy parents out there. Okay, these are zebra finches, and they have a very distinct, you can sort of see some of this is frequency mapped over here on this side. These are trills. These are little pop-like things, I guess. And then they have these sort of modulated tones that they make. This is actually the call that the zebra finch would make. It would make several of these individual ones, and then a series of longer stereotyped sounds. And the way the zebra finches learn this is by listening at a particular time, which is from, I guess, 20 or 19 days to about 50 days. Then they repeat it for a while, not necessarily hearing it again. And then when they actually start being in an environment where they're going to use it, i.e. mating time, they start to, sorry, I just lost my point. Then they start to make the final sounds. And after this point, it's called crystallized because it locks in, it doesn't change anymore. But if you try to influence their call during the period after acquisition, during sort of when their brain has already taken in the input, there will not be any significant impact on the final outcome. So you could give a completely different pattern in this period right here, and it probably would not impact the final mating call. Whereas if you give it about 20 times here, any point by a model bird, it will train to that. And so vision systems are used as a tool for analysis, in part because it's something that we can manipulate quite a bit easier than what sounds animals get exposed to, or we can't eliminate all sounds very easily. It can take out years, but it's a bit more difficult. Vision is easier. We can limit, say, one eye. Most animals and test models have two eyes, so this provides a way to start to tease things apart. So this is a very common model with some of the early experiments. So input's easy to modulate. You close one eye or you close it for a short time with sutures or other mechanical means. You have two segregated inputs that are present, and you can say, okay, well, if I stimulate one eye or the other, what do I get? You can change the stimuli after the experiment, and then you can look at the connections from retina to cortex using tracers. So this is another technique that gets used. And additional input is also possible. This is where we add function and see what happens. This is the genicular nucleus of the thalamus over here, showing, I think, ipsilateral and contralateral contributions. I think the ipsi is dark, but I've got to check back on the diagram. And then this is the actual projection onto layer 4 of the pelican cortex. And this mapping here is, I think this is macaque, but I'm not sure. Could also, no, maybe it looks like a monkey brain, so it must be macaque. This here shows ipsilateral and contralateral inputs that have been traced out from the eye. So the original tracer injection is proline into an eye, it goes back to the genicular nucleus, and then it jumps the synapse, goes on to the next neuron, and goes back to the cortex. And it will hold true to where the connections are. So it's one way of mapping it out without Golgi staining and all that. In fact, functionally, if you do just staining of the genicular nucleus, there's no real particular stain that will say what's one eye versus another. So this is a very useful functional approach to do anterograde tracing. Sorry. Okay, so this is the proline jumping across. It basically gets incorporated into proteins in the initial neuron in the retina, and projects the lateral genicular nucleus, which then projects to the cortex. And then the protein basically incorporates the proline. The proline's radioactive. It protein products and proteins themselves get released, get absorbed, and then the chain goes on. And it's going to be a sort of a proportion thing. So some of this is going to leak out and go other places, and that's going to lead to some degradation of the signal, but most of it should go in the right way. At least that's been the result of a lot of experiments. Okay, effect of monocular deprivation. This is in a cat model. And what you've done here, let me explain the scale on the bottom. These are, I think ipsilateral is all the way in 7, if I remember right. Yeah, so they've looked at all the neurons throughout the cortex in the, basically the visual cortex of the cat, and seen how many respond to stimulus from the contralateral eye versus the ipsilateral eye versus some graded response. So responding to both would be 4. You respond equally to inputs from either eye. So this is a normal cat, and there are significant numbers of neurons that respond to both eyes. In layer 4, obviously that's different, but this is the whole cortex. Then when you cover one eye, you get a lot of ipsilateral response, and you get minimal response to the contralateral eye, which has been closed. And when you do this later in life, after I think it's 12 weeks, 6 weeks, birth, yeah, I think it's 12 weeks, then you get much more of a normal bell-shaped curve. It is kind of suppressed, and I didn't see a lot of remarks about that in sort of textbook, but it makes me wonder, well, what overall impact has the deprivation had on the connections? Are they generally weaker, or what's happened because of that? But the shape nevertheless indicates that there hasn't been a selective pruning of pathways from one eye. Okay, and then they narrowed down this little period. We were talking about weeks before. It's actually at the end of the 4th full week of life for these cats, the kittens at that point. And it's about a 6-day interval that seems to have the biggest impact. So you can get full ipsilateral stimulation with just blocking things about 6 days right before the first one. Okay, now with the macaque monkey, part of the pathways are actually determined before the monkey is born. So it's not quite as clean as the cat and the frog, I guess. Here, this is a normal visual cortex, and this is one that has been partially deprived. And even though it's been deprived, you still see both dark and light areas, indicating that there is some still input from the deprived eye, even though that eye was deprived at birth. All right, and this is showing the arborization from the lateral nucleus endings in the visual cortex, so going from the thalamus out to the cortex in non-deprived and deprived eyes. And this is short-term deprivation and long-term deprivation. It's a similar effect. Okay, Hebb's postulate is one of the big ideas that comes out of all this. And this was trying to clarify a little bit how things get selected and how particular pathways are enhanced. But basically, the idea was that patterns that enhance each other's signal are favorable. And then when you get sort of chaotic noise, you don't get the same kind of feedback, and those patterns are sort of lost. In other words, connections are lost that don't make sense to the receiving nerve cell. And there may be some competition here for some kind of little reward for connection, if you want, or some kind of a growth factor. The similar patterns tend to give are given less weight and are eventually pruned. An example of this is amblyopic eye losses. So basically, if the vision from the two eyes is not in sync due to one eye having some differential muscular alignment, then eventually the brain will actually limit the input from that eye, and the eye may have limited or no input at all in some individuals after, I think it was a couple of years. Then that's the outcome of that for a lot of individuals. And it's the human example where we have some pruning, where because the affected eye has sort of input that is at odds with actually seen in the good eye, tends to get pruned. So here's an example, here's sort of a diagram of what might be happening and what has been shown to happen sometimes. It's a little hard to see, but you can see a pattern generated from these three neurons, which is a 3, 2, 1. And then there's some other neurons that would be looking at, say, the same stimulus with a different perspective, say, from the opposite eye. And you see a different signaling pattern, 1, 2, 1, 1. And because these don't line up with the majority input, they actually could be pruned. That's the idea. So there's an inhibition going on. Okay, so adding an input was something that was tried using frogs. So Xenophus was transplanted with an additional eye on the same side as one of the eyes. And then they looked at tracer to see what happened in the cortex. Now, Xenophus is a bit different than macaque and bats in that one eye goes to one. Yeah, actually, this is the geniculate. Sorry. So one eye goes to one side of the geniculate, and the other guy's eye goes to the other. And when you add the extra eye, these two are actually competing to try to influence the cortex, and they end up with sort of visual columns in the geniculate with competing eyes. And the other eye goes to the other side. And this is actually the tracer that they injected and showed that if you inject the tracer to this eye, it would generate those patterns. It would actually do the same thing with this eye, and then the other eye would light up the opposite side. It just proves add-on function. So cats with strabismus, where you have one eye that's out, that's not aligned with the other eye. And what happens is that the distribution that you see in the cortex is actually much enhanced. So instead of having a distribution showing stimuli from both eyes are somewhat Gaussian or distributed through differential sort of stimulus for each of the nerve cells, you get divergence, and the optic columns would actually extend not just through layer 4, but you basically have a column here, and you would have the same thing going up into the higher cortex.
I'm a little confused about this. Is like is the 1 to 7 the layer of the cortex?
No, it's tempting to think about that, but no, it's not. One is a nerve cell that's responding to only contralateral input, and it could be anywhere in the cortex. And then 7 is only ipsilateral input is what's driving that out. So there's no eyes, no neurons in this eye's cortex that are responding to both. Okay, now the molecular mechanisms here, this shows basically calcium doing calcium type stuff with the, I think it's ERK, endoplasmic receptor mediated kinases going into the nucleus, doing their stuff with Krebs. Krebs works with cyclic GMP, is activated by cyclic GMP, and then it drives transcription factors. Now, one of the things it could drive is BDNP, a brain-derived neurotrophic factor, and that's what's thought to go out and stimulate inhibition. And this was one of the big experiments on the molecular level to try to show that. This is one of the classic experiments. This is an embryo with nerve tissue culture, and this is up here with control medium, and you add snake venom, which is basically conotoxin, something like that, and you're inhibiting all the acetylcholine type activity, and you get some sprouting. And this right here is with purified BDNF, and I get more elaborate sprouting. So this is kind of an example of how the growth and activity of axons can be modulated by something as specific as BDNF. This was done by an Italian neurologist, neuroscientist. She passed away, I guess, about 6 years ago. Very influential woman in science. Some of her experiments lasted quite some time, or had an impact for quite some time. All right, other systems that might be involved, or other examples of model systems. Owls with disrupted hearing, you can basically take out an ear. I don't know that there are cortical columns in the same way with the owls. I'm not aware of that data, but there are barrels for rats. There are basically there's not stripes, but columns can set up in many different parts of the cortex, including somatosensory, visual, obviously, and some mechanical sensory areas. Monkey babies with the bonding to surrogate mom is another example. One of the experiments was to try to bond the monkeys to particular types of surrogate moms. One had sort of it was very raw, metallic, with some chicken wire around it. It was a model of a monkey with appropriate food, and the babies did, even though there was food there, preferred to hang on to a mother that was softer, with some terry cloth, and more supportive in sort of a tactual way.
They have like warmth too. They like heat.
Yeah, so that also had some apparent acid role in generating the behaviors, but it was used to tease that apart a little bit. Olfaction in rats, I mentioned this a little bit earlier. Mom sent imprints to children, and they will look for that in their mating behaviors later on in life. And then sheep, again, the mom may, and this is actually the mom imprinting, which is a little weird, but she will recognize only her own lambs after, I think it's a 4-hour period. It's a very brief interval after birth. So if the baby is separated from her for more than 4 hours, she won't recognize the baby. That's kind of important to the sheep on it. Okay, cortical development is there's a critical period that seems to come out of this, at least for some connections in some systems. The outcome depends on what inputs are sort of present, and on what effect your inputs have on the cortex, because you can manipulate these. If you can also apparently change some of the neurotransmitters, maybe some of the second messengers at the cellular level, and have some impact on not only whether it happens, whether the connection happens, but the timing. That's kind of important point. So when they're doing BDNF experiments, the timing of some of these connections to the optic or the visual cortex, it changes because of changes to the concentrations of these trophic factors. So absence of input also affects development, and so something that's kind of interesting from a developmental biology point of view, as well as neurological development. There's still ongoing work on this. We don't know everything. I think is another example of just language and how we develop that. Trying to map that out with functional MRI and all that is kind of complicated. So sometimes the system becomes so complicated that it's just difficult to set up the experiments. And then we're also dealing with humans, and that limits the practical things we can do in setting up experiments. Anyway, that's, I think, the last slide. So, you're welcome. Lunchtime?
Yeah, you can just close the PowerPoint off.
Okay. Oh, we lost all the students.
Yeah, they have medicine lecture.
They lost their interest.
Yeah.
I kind of figured.
No, I'm joking.